The question around personalization of follicular lymphoma therapy links up directly with the Genomics England program, where we are going beyond, chronic lymphocytic leukemia in the pilot phase of the Genomics England program. Really depends on the availability of appropriate samples from the patients. As far as follicular lymphoma is concerned, there have been many studies in the last few years, both looking at clinical measures for outcome and predictors of outcome. There have been molecular markers that have been added to the clinical markers, and then more recently a study was published looking at gene expression profiling in follicular lymphoma. Interestingly, a very comprehensive study that was published end of last year, looking at 1,001 diffuse large B-cell lymphoma cases, I showed that the combination of clinical parameters with DNA and genomic markers and gene expression markers has a much better predictive value than any of the others in isolation. And so my prediction is that where this will all end in the end is doing what we call multi-omics approach of using a mixture of DNA markers, gene expression markers, maybe even some protein markers, and clinical parameters to identify patients who will have a short time to first treatment or very short time to next relapse. And that applies to follicular lymphoma. It's a nice example, and diffuse large B-cell lymphoma, I'm sure it'll apply to all the other diseases as well. Now, with regards to the Genomics England program, of course, that is the first step in this multi-omic direction. However, the practicalities and the NHS transformation that is required are proven very challenging. So specifically for the haematology program, or the hemato-oncology program, I should say, is the availability of good, high-quality germline specimen. And we really need those in order to identify the mutations that are acquired in the leukemia, in the lymphoma, and not just something that is present in the germline of the patient. So we do need those samples, but very often they are contaminated by tumor. And so this is the specific challenge that we are facing, and we've done a lot of experiments now and are still doing a lot of experiments to try and identify what the optimal germline source is for the different patient cohorts. So to cut long story short, about 200 patients have now been recruited with either lymphomas or leukemias. There will be, I think I can say this, commissioned whole genome sequencing for patients with hematological malignancies. So this will become part of routine NHS care, but the validation efforts and the germline efforts are still ongoing. So it's not a test that will be available on the NHS tomorrow. It's something that will take a little bit more time in the next year, I would think.